All right, so I think just to start off this morning, so my name is Ronan. I'm at the National Cancer Institute. I work in the Division of Cancer Treatment and Diagnosis, and we more or less act as the drug development arm of the NCI, so we sponsor 900 clinical trials around the country. We also work collaboratively with other organizations that you might be familiar with in the big data space, like the Surveillance Research Program that publishes SEER, the Healthcare Delivery Research Program in the Division of Cancer Control and Population Sciences, both of those. And we've been thinking about different ways that we might be able to engage with stakeholders and leaders in this field so that we can better understand where we go next, how we help patients, and pay really close attention to essentially what is a very fast-moving endeavor and effort. And so it's really gratifying to be able to introduce the panel. I'll just mention we have Amy Abernathy from Flatiron, Andrew Norton here from CODA, Elizabeth Christilis from Coronet and also representing the University of Iowa, Joe Hank here from Upton Labs and the Mayo Clinic, Larry Kushi from Kaiser Permanente and the Cancer Research Network, which was an NCI-sponsored effort, and Nancy Dreyer from IQVIA. And I will start with Amy and go through the list to just have you introduce yourselves, just a couple of minutes, and sort of talk about your role or your interest, I guess, in the project, and we'll probably build some discussion points from there.
So, hi, my name is Amy Abernathy. I am the Chief Medical and Chief Scientific Officer at Flatiron Health in New York City, which is a cancer software and data company focused especially in the real-world evidence space. Prior to that, I was at Duke for a very long time. I was a professor of medicine and an oncologist and ran the Center for Learning Healthcare. Why am I excited about this project? So we know, or we think we know, that real-world evidence is going to help us understand how to put information about the drugs that we have, the interventions, healthcare in general, in context of the patients who sit in front of us every day. But if we're going to use real-world evidence, then we need to understand how to make sense of the information of the data sitting in front of us. And I think this project is a remarkable project to start to put different types of data sets and information generated from those data sets in context so we can think about how we use them. So I think one of the reasons I'm excited about this is we actually see this example now playing out in 6 different data sets, which gives us both the understanding of the breadth of the kind of information as well as remarkable similarity of information around a specific project.
Thanks. Andrew?
Hi, I'm Andrew Norton. I'm the Chief Medical Officer at CODA Healthcare. And CODA is also a company in the real-world data and analytics space focused on cancer. Prior to being at CODA, I was at IBM Watson Health, leading up some oncology and genomics efforts there. And before that, spent the bulk of my career as a neuro-oncologist and clinical trialist at Dana-Farber in Boston. I share much of Amy's enthusiasm for this work, and it's really been a tremendous collaboration. I think collaboration is going to be a really key thematic element here, in that we can do a lot more when we work together than when we work in our silos. We at CODA approach real-world data from the perspective that physicians are spending enormous amounts of time entering information into electronic systems to the point that they can hardly find time in their day anymore to look patients in the eye, which is something that every oncologist and patient laments. And yet, to date, we've not been able to make much, to learn much, to drive knowledge forward much from that information. And so we're looking to change that. In order to change that, we need to have some agreed upon approaches that describe how we actually use and work with that data. And I think this is a great step forward in the direction of being able to.
Elizabeth?
Hi, I'm Betsy Christilis. I am a professor and department chair for the Department of Epidemiology at the University of Iowa in the College of Public Health. I have the privilege of being a co-PI for the Cancer Collaborative Research Group with PCORNet. And the University of Iowa is one of the, about, I think, 80 sites that participate in PCORNet. I'm our local site PI. I've also got the privilege of sharing data from a pilot project that PCORNet sponsored to examine the utility and validity of PCORNet data resources for harnessing real-world information about cancer therapies and outcomes. So in that pilot project, we have 11 sites, 11 of the 80 sites participating, and those are the data that are contributed. And I'm very thrilled that those 11 partners were willing to share their data in such a rapid time. And I guess one of the things that excite me most about this, as an epidemiologist, replication is our friend, and this is just this amazing ability to replicate rapidly, and I think this has been a thrilling experience.
Joe Hank, Optum Labs. Optum Labs, in case you don't know, is a research collaborative started a little over 5 years ago between Optum, the United Health Group company, and Mayo Clinic. And so I should recognize Aaron Mansfield. It's his anniversary. He's not here. He's the oncologist leading the study, of which he allowed me to borrow the data. So speaking of collaboration, it was great that he let me use data from his study to participate in the panel that originally Jeff approached us with. So I'm going to second Betsy. I was very impressed how quickly things happened, and prior to my stint at Optum Labs, I was in consulting, so things happened a little quicker there. But I've also, as an academic, some things happened much slower there. So I think how quickly people were able to bring data together and how much data actually exists in ready-to-use form was quite impressive. I'm a methodologist by training, statistics and econometrics, so a little bit of a fish out of water. That's how I attach myself to a research oncologist for some clinical expertise. But some of the stuff that I think are short list are a lot of methodological, not access to some of the data was quick enough. It's how to analyze the data and how to improve the data. So it's I hope we move forward. Speaking of trying to do your job for you, it would be nice if we could stick together.
Hi, good morning. I'm Larry Kushi. I'm at Kaiser Permanente in Northern California, based in Oakland, California. My training and background is in epidemiology, like Betsy. And as Rod mentioned, I've led an NCI-sponsored grant called the Cancer Research Network over the last half dozen years, been involved in it for longer. And the Cancer Research Network, I'll just describe, is basically a consortium of a dozen healthcare systems like Kaiser Permanente in Northern California, originally HMO model community providers. And we've existed since the early 2000s, 1999, actually when the grant was first funded. But part of the purpose of the Cancer Research Network is basically to support doing research around cancer in these healthcare settings. And so one of the first things that we had developed early on, before I was involved, so it's not my idea, but I'll lay partial claim to it in terms of its use, is the development of a common data model called virtual data warehouse, in which data are extracted from the clinical and administrative data that each of these healthcare systems have. And so the data that we provided here are based on the virtual data warehouse. Now, the virtual data warehouse does not include every single data element that is actually of potential use or relevance, even to the questions that we were addressing here. But it does provide an opportunity for being able to look at certain types of questions in a relatively rapid fashion. So, like the previous panelists, I think there's a great example and a great opportunity to learn from each other, particularly in this cancer space, because of so much change that's happening in clinical care and all the things that we can really take advantage of what is happening in the real world to really develop learn the healthcare system in the big sense of the word, not just what may be happening in Kaiser or something like that, but actually how can we impact care and really improve sort of cancer outcomes for everybody across the nation. So I'm really gratified to be part of this panel and part of this project. I think it's been a great collaboration so far, and I look forward to seeing what our next steps are going to be.
Terrific. Good morning, everybody. My name is Nancy Dreyer. I'm the Chief Scientific Officer at IQVIA, which is a human data science company. And if you never heard of IQVIA, that's because we only took this name about a year ago, and it represents the merger of IMS Health and Quintiles. And I'm a practicing epidemiologist and pleased to be in the company of so many like me. I've been doing this for more than 30 years, working with primary data collection, with health insurance claims, with electronic health records. And to me, what's really exciting about this project is, first of all, kudos to Friends of Cancer Research for bringing us all together and for driving this through. And what you see here today was done in about 2 months. And it's not exactly 100% replication of exactly the same methods, despite our best intent to do so. But we took about 2 months to try to replicate this protocol with some variation and some fine-tuning. We've got claims data, we've got electronic health records. Some of us had time to do natural language processing, most of us didn't. So this isn't the very best of what can be done. But that said, the amazing congruence between what we found and the similarities with trial data, I think, is an amazing message that's important to take home. We got the same directionality, we got in the same ballpark. If you want to argue to number of decimal points, we can certainly do that. But the finding for patients, for physicians, for public health is, I think, is very important out of this research. So my sincere thanks to Friends of Cancer Research.
Thank you. Thanks to all the panelists, and actually thanks also to Friends of Cancer Research for putting everything together. And also thanks for being very succinct. This was one of the fastest introductions I've seen from a very distinguished panel in recent memory. So just to, I guess, hit on some of these themes that we were interested in developing further. In the process of putting this together, did anyone feel that they actually developed more of an understanding of what the potential needs or gaps were from their own data sources and how we might be able to address them going forward? And I guess I can just mention we hit a little bit of a sweet spot. This is a very fast-moving field. Even just at, and when I say this, for me, I'm talking about the development of immunotherapy in lung cancer specifically. I actually also state that really big data in oncology is actually a very fast-moving field as well. But just at AACR and ASCO this past year, we potentially developed new standards of care for the use of the immune checkpoint inhibitors in lung cancer and advanced lung cancer in the past several months. So we sort of hit a spot where you had approvals that were available to patients in the real-world setting from 2015. Early 2015 was when nivolumab got approved. I think mid-2015 was when pembrolizumab got approved in the advanced settings for patients with non-small cell lung cancer. And now we've got a different, essentially, taste of where these drugs might actually be used in the future. So I guess the question would be, do you feel that we're in a good spot to answer questions like this going forward, or what other needs or gaps did you develop or identify along the way? Maybe I'll start with Larry.
So if you recall from the slides that Jeff showed, comparing some of the results of, in one of the last tables, there were only a couple of our data partners who could include information on progression. So that's one big thing, I will say, that being able to identify progression in a structured manner is, at this point, extremely difficult to do. So that's, I think, one big need. Another big need, which was not shown but is demonstrated in the tables, is that only some of us could also identify the actual tests and the results of those tests that would result in actual use of these immune checkpoint inhibitors. So the PD-1, PD-L1 test, for example, the fact that the test was done and the results of those tests are not readily available. Those, explicitly in this context, and the questions that we were all jointly addressing, I think are clear limitations in terms of the way electronic health records and the data that are readily available are currently able to be used to address these questions. So I think those are a couple areas where we clearly could use better information. Another, which I think people generally recognize, is that death data is sort of lags in terms of when they become available. So with new approvals, as Eli just mentioned, the ability to address those in the survival context, even if you were talking about survival over the next year or something like that, is limited because the ability to link up with death data sources lags substantially from the electronic health record data, for example. So those are three examples, I think, that there could be real data improvement. I could go into more details about some of those things, but I think that those are clear ways where, if we collectively, not just amongst six data partners up here, but more broadly with many of the other efforts that are ongoing in this general space, we're able to get access to structured data, I think that would really be helpful. Betsy?
If you don't mind, I just wanted to follow up on, I agree exactly with all three of those points. Those would have been the three that I would have called out. But two of those I just wanted to follow just a little bit more about. So with respect to test results, and I think this also relates to Nancy's comment, there's so much more that we know we can do with these same data sources, but in the space of 2 months we really couldn't. Some of us had curated progression up and available, others of us didn't, and that would have involved some chart abstraction. But my point about the test results is that we had a separate small study that was examining completeness of the test result data if you do look at the EHR for these therapies. And at least in our academic medical centers, it's an open question as to whether that would be there. And we found that, yes, indeed, it is there. Sometimes in fairly specific locations that might have some potential for structured extraction. Other times probably more in ancillary PDFs or text notes that would need some NLP. But the information is there, so that's very positive. And then regarding the death data, we had an opportunity to experiment a little bit with sites that had different sources of death information, and we learned from that the incredible importance of the external sources of death data, such as state death data or Social Security death data, to complement what the local systems were extracting in their daily course of business.
I would add on to that last point about death data. We've looked in our network and found that the local systems are capturing death data. Now this is in where we are maybe 10% of the time. And I realize it likely varies by system, but I worked at a large academic cancer center. There was a major effort for years to get physicians to simply note in the record when they received a call from a hospice agency that the patient had passed away, and compliance with that was extraordinarily poor. And so I think it varies by system, but that's hugely problematic, and we have found. We've had some good luck and are now incorporating obituary data. But Amy and I talked in the limo last night, or it was not a limo, it was a car. At any rate, we spoke a bicycle. Yeah. Right. We spoke about the fact that this is a critical need and that it would really be quite valuable to this entire effort if there were a centralized, up-to-date death data repository, and frankly, that would drive the field forward in a really remarkable way. On the progression side, it's true that only some of us have access to progression data at all, but I don't know that we all define progression the same way. There's no protocol governing the way progression is determined in the real world. There are no standardized assessment time points, and we don't follow criteria. So it might be interesting to talk about how we, CODA and we, Flatiron, define progression differently, but undoubtedly this is an issue, and the field would benefit by standardizing the definition.
Before we end, can I just add one more thing on depth? I know there's lots more to talk about. You're probably sick of hearing about depth, but you asked about what things could be changed. So we've talked, we do have a national death index. It's phenomenal, but it's slow. So even if we can link to it, we need to wait 12 to 18 months to make that linkage. And we've talked about the fact that there are a lot of good reasons why death isn't entered into a record or a health insurance claims data. But there are opportunities to link with state death registries, with credit bureaus. They actually know if you're dead. But I think a take-home message for everybody is that people are becoming so concerned about privacy. And I'm not just talking about private credit bureaus. I'm talking about Medicare, Medicaid, data linkage, and the way to do that in a way that respects privacy and doesn't violate the laws of small numbers and can't be used for re-identification. But we know how to do that. But I think there's growing caution in this country, as in Europe, about the desirability of sharing those data. And we need to raise awareness how important those linkages are for efforts like this. Because the system is such that most people aren't going to call the doctor to say, my husband passed away. They got other things on their mind. So we do need to be able to make those linkages, and they're going to be important. We're very sensitive with our death data. We know, at least in our experience, we're pretty sure when we say someone's dead that they're dead, but not very specific. So if we don't know that they're dead, it's a little bit, it's not clear.
So I wanted to follow up on that, and there's 3 observations that I had as it related to some of these. So one of the things you brought up a lot is that the use case chosen reflects a rapidly changing space in cancer care, and understanding the underlying clinical context of what's going on and how does that interrelate with what is the related characteristics of the data set as well as the findings, to me was one of the things that really struck me when I was looking at the results. So one of the things is that we've shifted our use of immuno-oncology agents towards the first line setting. And what you saw was, depending on the recency of the data set, you saw different proportions of first line usage within the data set, which then ultimately also influences what the data set can tell you in terms of outcomes and other things. And so understanding the underlying clinical context, how the timing of when the data were collected and processed and analyzed, and what that means in the context of what was going on clinically becomes really important as you interpret findings. And that was one of the things that I had as a takeaway in this. That also goes along with how do we think about things like mortality data, because depending on the mortality data source, if you need to wait for the source to essentially mature enough that you know that it's complete, that actually influences the timing of the data sets. And I could, you can follow that as you look through the results. So that was one of the things I had as a takeaway. The second takeaway that I had, and when we were doing this project, there was one point or another where I sort of had a panic attack. Those of you at Flatiron know exactly the night that this happened. When I was looking at some of the results and said, no, that does not make sense. And what happened was we looked at the time to treatment discontinuation and the real-world progression results, and the correlations with overall survival were in favor of time to treatment discontinuation, which I've never seen before. It just turns out that one of the changing areas in clinical care and immuno-oncology agents is that, in fact, we're starting to see that there is likely a survival advantage to treating past progression, so oncologists are starting to treat past progression. So what does that do? It basically tightens the correlation for time to treatment discontinuation and overall survival. So the importance of taking findings that don't make sense and then drilling down to try and figure out what's going on is a really important feature as we start to work with these data sets. And that actually takes me to my third one, which was my kind of aha listening to this and thinking about what happened at Flatiron. It's that partnership across data sets, across understanding, but also very specifically across all the people who are working on getting to the answer that really made a huge difference. You needed clinicians, you needed experts, statisticians, and epidemiologists. You need an understanding of what's going on right now in the current clinical context to pull all this together. And to try and do that with a 2-month timeline means that you actually need a communication structure where everybody understands how to talk to each other.
Just to bring up, since we're talking a little bit about the progression-free survival, there's obviously been some work done at the FDA that sometimes these particular surrogate endpoints don't necessarily correlate with overall survival. But at least one of the data sets that happened to be, I think data set D, if I'm not mistaken, had a rather tight correlation with overall survival or progression-free survival, if I'm not mistaken, with a rather small confidence interval. Is that, can anyone who would like to comment on that, is there an effect of censoring that's going on in terms of the ability to say that you have some of the data for both those particular endpoints and that's why they happen to correlate well? Or I'm actually just asking out of curiosity, I don't have an answer.
So maybe I'll take this one. Yeah, you're right. There was a pretty tight correlation between overall survival and progression-free survival. When I think of that, first of all, when we calculate progression-free survival, that calculation includes survival, right? So some of this is the correlation between survival and progression-free survival in terms of OS and PFS calculations. In fact, in a disease like lung cancer, where there's more mortality in the period of time observed, what you end up with then is a higher likelihood of deaths and therefore a tighter correlation. In another disease where you have fewer deaths over the period of time, the correlation is likely to be lower. So in one of the things that you're seeing here is a tight correlation between PFS and OS that's related to the disease itself. The second is that tight confidence intervals often belie ultimately your ability to talk about answers with a level of precision, which is partly the features of the data set as well as the size of the data set and the features of the variable itself. And then I think in a disease like lung cancer, progression is more likely to be a surrogate measure, and we've seen that in clinical trials as opposed to other diseases where something like progression may not be a surrogate for survival because of ultimately lots of issues such as multiple lines of therapy and other things going on.
There were only 2 data sets that had progression, and one of them was much smaller than the other, and therefore had a much broader confidence interval.
So we have large and small. We also have variations. I think it's just important to remember the way we executed this. This exercise made me appreciate some of the work that Sentinel has done in the sense that they share code and they send out executable programs, which is easier when you have a structured data format, and I'm not suggesting we all need to do that. But sharing code and just even the way that we looked, the way we did censoring and the like. So I'm not sure we constructed our cohorts exactly the same, nor use the same code. So but your point about the size of the data sets driving the confidence intervals, of course, true no matter what.
And I really second that. The ability to have consistency in how we calculate these things, consistent business rules, sharing code, I think was one of the other critical learnings here.
Yeah, that's definitely something that from some of our other CRN projects, being able to share code, etc., really does help to result in similar definitions, at least of some of the key variables, such as who actually has advanced small cell lung cancer, which is solving the codes that we're talking about, or whatever. Things like that really do help to result in some sort of consistency, which we didn't really do, but we still have pretty amazing consistent results across the 6 data sets here. So that, I think, since we all obviously communicated with each other, I think that was really helpful. I wanted to make a point about the lack of information in structured format of the actual tests that ultimately result in the use of these therapies. Because if you have that information, if you have the information on were the tests ordered and carried out and what were the results of those tests, then you can start to address questions like, okay, what's the therapy that was given? Did it align with the test results? Is there off-label use, so to speak, in terms of what the test results look like? And those are questions that we could, in theory, answer if we had that information. and some of our data sets did, and some of them did not. And so PDF documents in clinical notes, whatever, that type of thing. So it's not readily available to people who want to look at these quantitatively. The interesting thing is that usually these data from where they originate, the actual labs that do these tests, they are available in structured format in those settings. And so being able to link up that original structured data with the EMR data that now exists in clinical practice, that will go a long way towards being able to do that. So being able to bridge that gap rather than perhaps trying to do NLP and trying to sort of re-enter data that are sitting in PDF documents attached to the EMRs. I think that if we could do that, that would help bring in more sort of data sources, but sort of really address the big data sort of ecosystem in a way that could really help improve care.
That latter point really rings true with me. I mean, I know that we have built an expensive and very time-consuming human enterprise of pulling out the very data you're talking about, and it's incredibly frustrating knowing how many man and woman hours are being spent on this when, in fact, it exists somewhere as structured data that could easily be imported.
Yeah, I know you want to move on to the next question, but I want to add something. I'd like an opportunity. So the one thing, and this was only a pilot, like Nancy said, a couple months of real work and across 6 groups, so there's some inefficiencies. But one point is that there's a host of questions on why results differed, and the data largely exists in some format. So if the group does continue, I bet we could learn a lot about what the differences are. And one point about data, I was thinking data generating mechanisms, and we didn't talk a lot about the data sources, and we probably won't, but in the administrative claims system, a lot of that data is generated from payer information, and it's a system, and part of that system is prior authorization, especially for IOs. And so some of the questions clinically or from a provider perspective that might be relevant, but perhaps they're solved by some of the prior authorization approval systems that are in place. So anyway, my point is not to educate everyone on prior auth, it is to say that without understanding the data generating mechanism, it's a little difficult to analyze. And that might explain some of the differences.
I just wanted to take the moment to make one observation. So we're talking about problems with underlying data and sort of issues that we ran into. As Betsy said, replication is our friend. One of the things I found remarkable was that it didn't matter what data set, age did not predict survival, right? So it being over or under 75, you saw exactly the same outcomes across all the different data sets. So whereas it might be hard to say the overall survival is exactly 10 months or whatever you're going to say, it really is the ability to use these kinds of projects, data sets, work to start to make clinical observations that we can then put back into our clinical practice. And one of the ones that was just to me remarkable was early age.
Yeah, just getting back to one of the comments that I think Andrew made is that there is no real, I guess, benefit for the provider potentially in reporting into the system the death. One could actually argue that that's potentially an incentive problem systemically, that we aren't really being incentivized to provide that information into the medical record in any way. And so, we care a lot about death in oncology for a lot of reasons. It's a gold standard that we can use for understanding patient benefit. Extending life is clearly in our mission statement, I think, as oncologists. So what we're trying to do, or I guess from this panel, from this meeting, is figure out how we can alter our system somewhat, or at least make proposals as to how we might be able to alter our system in order to make it easier going forward. And so I'm wondering, should we actually provide some sort of financial benefit to health systems that are reporting death into their medical record? Should that be a requirement, when the information is actually available, like a mandate, or are there other things that we might actually be able to use to incentivize better record keeping so that we can move forward.
As I do a lot of work in patient registries, so and in working with registries, somebody always knows. There's value not just for healthcare and healthcare-related research, but there's value for all sorts of reasons, financial and otherwise. So I think I would put this on a central body to help ensure that data is captured and shared and done so in a timely way because it benefits us in a lot of different ways. And obviously we have to solve the privacy concerns.
Well, HIPAA doesn't apply to dead people, as far as I know.
no, but
Privacy
No, actually, privacy probably doesn't either in some ways, but HIPAA
There's
certainly
no
does.
privacy at the fact of death, but I just want to add that we've done some experiments where we actually have patient consent for the linkage, and still the partners won't link because they're worried about privacy. What's that about? I mean, if you're willing to allow someone to check, it seems to me that there should be an obligation, whether it's a government or a private agency, to honor that.
So I just wanted to add two pieces. One is I've been really pushing the National Academy of Medicine to think about this and try and push it forward. So I think from a policy perspective, we should figure out where to concentrate our voices to be able to try and push this conversation forward. The second is I completely agree about linkage, and you can amalgamate across multiple data sets, and we have a paper in Health Services Research 2 months ago looking at amalgamation and then being able to test sensitivity and specificity. But there's actually a third thing, which is that we should be doing the experiments to how do we improve the collection of reliable death data as a part of day-to-day healthcare. Because that's going to be at least one of the data sets that links in. And here's one of the things that we learned. It turns out that clinics who systematically send condolence cards as a part of essentially closure for the clinical staff know when patients have died. So if you put into the clinical workflow the sending of condolence cards, it's actually good for the staff, it's good for the family, and it also improves death data. So there are things that you can do to systematically improve data collection.
So one point I wanted to make regarding incentivizing putting information into EMR or whatever is that, as Andrew mentioned in his introduction, providers are already overburdened with documenting things. And from a healthcare system perspective, not that I speak for Kaiser Permanente or whatever, as a healthcare operations leader or anything like that, but a lot of these things that are incentivized, are related to quality of care, HEDIS measures, et cetera, that type of thing, and so those things do get documented really well if there are incentives. But I think that more broadly speaking, trying to incentivize for specific areas really does require really thinking about what is the clinical workflow, who is the person who's actually going to be able to do this, etc. Basically along the point that Amy just made, in the sense that if you have a workflow related to sending out condolence cards, then that could really help with some data documentation. Who's actually going to do that and where is it going to fit in, and those types of things, I think, are sort of critical questions if you do want to try and develop sort of better documentation in any of these areas. I think an example is stage of disease in cancer. How is that documented in the routine clinical workflow? It's clearly there, and it's clearly drives treatment, etc. But on the other hand, it's in terms of the EHR, the electronic medical records, it's not really readily available in most settings. And I think many of us, probably either had it extracted or relied on tumor registries where it's required to be reported ultimately. But that's outside of the clinical workflow. So it's not in the electronic medical record per se. And so there are data sources. There's an example of linking from outside, in a sense, outside the clinical setting data set to pull in information like stage of disease, which is generally readily available thanks to SEER and sort of state registries. But on the other hand, it's not necessarily readily available if you're not linking up with those data sets.
One, just to shift a little bit off the provider data sources, is from the payer perspective, and I don't know if Medicare would ever consider this, but there is a lot of data collected, but not retained electronically. And I go back to the prior authorizations, especially for like IOs and treatments that are approved rather rapidly. There is prior authorization, so you do need to report stage, possibly histology, and other information. And to have that sit there in the stratosphere, so to speak, and not put in a database. And a lot of times the linkage is really, really simple. So Kaiser probably has this too, that for the enrollees, and in this situation, we didn't use this data we used just another data asset, but we did not link the two, but it would certainly be done. So just to recognize it's possible, and perhaps every large payer is doing it now, and not throw that data away and retain it for research would be valuable, and I would assume at a much lower price point than some of the active data collection.
That is a good point. I want to try to open up questions to the audience, just in case there might be a question over there. I can run a mic up front of the baseball cap.
My name is Nam Nguyen. I represent an international research organization for elderly cancer patients in over 100 countries in the world. My question to the panel is this: in your data set, how many are minority? Right? Because blacks have a more aggressive biology compared to Caucasian. The disease present at early age, localized disease at presentation. And on the other hand, on the other spectrum, Asian does not respond similar to Caucasian. The best example is Arasan, a medication for lung cancer, which is very effective in Asian, but not in Caucasian, unless they have a mutation. So does the survival differ among white, non-white, blacks, white, Asian? That's one we'd like to know.
I think that's a very good point, and we did have it's a little bit of an ethnic breakdown on this project. That actually something that was at least striking to me was that there probably were fewer, certainly than a population would suggest, African American participants in most of these particular data sets, and I wonder if we're having the same systemic situations where we're undercounting the broader population as we have in clinical trials. This is real-world evidence is actually supposed to deal with that issue, and yet it doesn't look like, at least in these data sets, that was particularly evident.
I was actually going to mention that as well. In spite of the variability that we do see across the different data sources, it did seem to me, at first, just at a simplistic sense, that we may be underrepresenting minorities. But if we think about the sheer magnitude of the sample size, so I think all told there were 13,000 patients, treated patients that were collectively included, which is 7 or 8-fold times the number of patients in the meta-analysis that was published this year for 5 trials. So there is some hope that we would be able to examine heterogeneity of treatment effect, treatment response across subgroups. I'm not sure the numbers are quite there, so I didn't do the math.
Yeah. I'll just say, I'm not certain if there's really underrepresentation per se in terms of the data sources, but it is possible that within each of these data sources, the minority populations that may actually end up being tested, being put on these therapies, that sort of thing, there may actually be practice differences that we can't, sort of detect specifically in terms of the way we did do this. I think some of us probably could start to look at those questions as well. But the data set, at least sort of broad characteristics, are in table 1, including the proportion of minorities. And so I think some of the variation in terms of the proportion of African American patients, for example, probably has to do with the settings that were being represented in whatever each of the data sets resulted.
Yeah, I think 100%, but I think it's actually a very good point, and it's something that speaks to some degree to the benefits of SEER and really truly population-based efforts that certainly the NCI is involved with. We also have projects that get published periodically on patterns of care, where essentially we take a sample of SEER patients and then look to see how they were treated and how well they did over time. The biggest problem, and I think that I think everyone's actually really quite aware of this, is that we have a huge time lag, because in order to do that kind of verification and to work with data sets that in fact are not electronic in many cases, or at least not as readily transferable as, let's say, Optum Labs or CODA or Flatiron or anywhere that participated in this project, throughout the whole population. You're talking about a big project just to use human abstractors and get that information verified.
But you're also looking at a sample of convenience. We were approached, we agreed to participate. If we went out to get even more diverse participation of databases in the United States, we would have the patients of interest. So this was opportunistic, as all efforts like these are. So there's no criticism, but this was handy and readily available. We can see the geographic diversity. We're seeing differences, and that's what you should see. So if we work to go to regions where we knew they had more diversity than in some of the ones represented, I think you could get those data short of doing a representative sample in the United States. So instead, pick geographic sampling.
Yeah, I think one.
Oh, sorry. Go ahead, Larry.
Sorry, just one last point. I think is that you mentioned that you represent an international group or something like that, and I think that it is really important to remember that we're all U.S.-based groups here, and where a lot of the data reside sort of right now in terms of sort of looking globally. And so being able to learn so that we can form not just our individual healthcare systems or the U.S. context, and also inform the broader global context, I think that's something that we really need to think about.
One quick point I just want to mention. Well, maybe should we take another question? Yeah, let's get to another question. I'll save it.
I have a question here. So I think this is a remarkable project. When we see the patient population, non-small cell lung cancer population across all 6 data sets have very similar demographic characteristics, but we do notice the clinical characteristics are very, at least there is some difference over there. And I noticed the data set C, at least 90%, 91% of patients were diagnosed at stage 4 compared to other data sets. And not surprisingly, that data set actually showed the lowest overall survival rate. I think, one thing I was wondering, and it's a question to the panel, we also look into methodology, and I do believe that the populations are not very balanced across the 6 population. Will you consider trying to do some advanced methodology like a proper matching, propensity score matching, and make sure that 6 data sets actually have very balanced, comparable population? Then look at overall survival and also time to next treatment. Because we usually don't have this kind of opportunity, and you have 6 data sets. If you can show when you apply the advanced methodology, you actually draw exactly the same conclusion with much smaller variance. That's a huge contribution to the real-world evidence research, because we do see in your overall survival, all the 5 endpoints, you see the variance, and some variance between 2 data sets is actually pretty great. That's my first question. Second question is, so I think Amy already mentioned that age is not a good predictor, and you notice so much from you repeating different research questions with different data sets. I think with the 6 data sets, it's really a vast opportunity for us to evaluate what confounding factors are truly there. Because you have 6 different data sets, would you consider the next step and really evaluate what are true confounding factors and what are actually not? That will help all the research community properly design their studies.
Well taken.
We're great. We move to one more question here.
Sure.
She was next. Okay, I apologize.
Hi. I have actually 2 questions. First, have you noticed the variability among the cohorts? And I know it hasn't, we don't know which cohort, which data set matches which cohort, but have you glean any hypothesis as to what might be driving the differences? For example, there's a variations in time to treatment discontinuation. That's pretty wide. Any idea what might be driving that? What's the cause of, have you drilled down into looking at the cause of treatment discontinuation? The second question had to do with the practice difference that you mentioned. When you're looking at the convergence of pivotal trial outcome data and real-world outcome data, you're looking at the difference between management that's very well controlled on trial versus, we all know, not the case in real world. What is your feeling in terms of that variability contributing to either overlap of the data versus, does this speak to lack of granularity of the data? Or are you suggesting perhaps there is a much of practice difference or that they don't contribute? I mean, I know where my sense is, but I'd like to hear your perspective.
Okay, maybe I'll start off. So I think both of these last comments point to the fact that there is in fact variation in what we see, despite the fact that we've also said that they're remarkably similar, given the fact that we really started this in earnest only about 2 months ago in terms of the actual analyses. We didn't share code. We talked about trying to identify the different important concepts in a common way, but we may not have necessarily. So the fact that the analyses themselves may be a little bit different, the fact that the populations are different, as we pointed out, in terms of some of the characteristics that you can see outlined in one of the tables, I think those are potentially sources of variation. We did not attempt to sort of adjust or to look at a similar population across all of these groups, like everyone who's stage 4, in this age range, in this particular, sort of gender or whatever. We didn't try and do that. We took the data that we had available in each of these data sets, and we came up with things that were pretty remarkably similar, given all that. Now, I think that we could potentially take it to the next step and say, okay, if we do adjust for, this characteristic, or standardize against some standard population, or something like that, then maybe we would see more similarity, maybe we'd see less similarity. We don't really know at this point because we haven't done that.
So I'm actually not asking about that point. I think it's great that you're looking at the real world data because you can't control from the real world. And I think that's one of the really big challenge of trying to do control study in the real world. It just doesn't work. But now that you have the data, it's about using the data the way it is and understanding what might be driving the difference. Right? And I don't think it's about narrowing your analysis to do the comparison. At least I think that takes away the power of the real world data and the diversity of the population you're looking at. But really asking much higher level questions to say, how can we understand, and I think intuitively we know practice difference makes a difference, contribute a difference in outcome. At least I believe that's the case. But how can we drill down which practice difference really makes a difference versus one that contributes less, because not everything is equal.
Yeah, and so that's exactly the next point that I was going to make, is that we didn't try and even explore things that we know should contribute to differences. Right? So once we do that and see if differences remain, then we can start to address some of these other questions. Or we can, obviously we can do it in parallel, and we don't necessarily have to do it sequentially or whatever. But I think that we didn't even explore, obvious sources of variation across these results. So we haven't even done that first step in a sense. And I totally agree that there could well be important sort of clinical practice variation, et cetera, that might result in differences in terms of some of these main outcomes. So that's something that could be potentially the next step in terms of really going down that way.
I think the big picture out of this is these are sources that go from very large to comparably small. But it's important to think of how the data came into these systems. Was it a commercial EHR system deployed in numerous sites who just happened to buy it and choose that one? Was it referring to a commercially defined population because of the types of insurance they had? I mean, all the characteristics of of that high level are so important. I think that's where you need to start. Are they academic centers? Are they community centers? The practices are different. So I think that we have a long way to go to understand between practices differences, where we first need to look at the health systems that are covered, why somebody was covered, and understand more about that population base of patients as well as practitioners.
We have another perspective.
Yeah, thanks. Vito from Merck. Two questions as the next steps. The first one is any plan to involve other data partners, particularly we talk about the other regions, like ex-US database partners to be part of this pilot project. Second question is regarding any thought about the safety endpoints, particularly we now see more and more combination therapies come up. Thank you.
Amy?
So the first one was about the other data partners, especially ex-US, and I think that's a really remarkable thought. You know, I think IQVIA, you really have international experience as you think about this. I don't know if you have any commentary, but the idea of being able to expand to other data partners is certainly high in our mind. But do you want to comment on ex-US?
Well, there are a lot of wonderful data sources around the world, and but the issues are the same as if they were sitting up here. We'd be facing the exact same thing: who's covered, why, who's included, what types of doctors, and what health system. As you brought up with the prior authorizations, there are reasons that patients get treated beyond whether the patient might likely benefit. So it depends on the physician's experience, the health systems, the availability, a number of issues. So I, of course, like all of us, I'm a fan of geographic variability and understanding those various factors to try to get the truth as what actually benefits patients in various situations. But the idea that we can drill down to subsets, whether they're ethnicity, geography, health system, that's the opportunity that the real world data brings to us.
And you know, to follow on, one of the key goals I think we were trying to get at in the beginning as we were working together is starting to figure out how to do this, and then the ability to expand further can move beyond. I mean, one of the examples I think you've heard a lot about is issues that we've started to run into related to mortality data. So working through that together here, that can start to be broadened. The other question was as it relates to safety endpoints, which explicitly within the context of this project talked a lot about efficacy endpoints, but the same principles apply, and starting to work through safety endpoints that we likely all have access to, and then safety endpoints that are more difficult would be important things to think about in subsequent projects.
All right, so.
Hello, my name is Wesley Platt. I'm from MITA Corporation. One comment and one question. The comment is that the National Death Index operated by the National Center for Health Statistics at